All right, welcome everyone. It's great to see everyone here and to welcome Al Kilman back to talk with us today. The invitation on behalf of myself and Andy Aguirre, so really happy to see him here. And today is our annual Krakoff Family Lecture. This lecture is now an endowed lecture. It's been going on for more than a dozen years. Let me just give you a little sense of the lecture, and then we'll introduce Alec and have him get started. But this is a lecture dedicated each year to pancreatic cancer, really inviting sort of the people that are luminaries in the field to come talk with us about their work and where they're headed. This is in honor of a number of people, but Sandra Krakoff, together with her family and the Coleman family, endowed this Krakoff Family Lecture initially in honor of Anna Maria Coleman to share advances in the field of pancreatic cancer. Prior to her passing in 2021, Sandy Krakoff was really a steadfast champion of Dana-Farber. She's a treasured member of the community and served as an institute trustee since 1995, and really supported progress not only in pancreatic cancer, but in many other cancers. In addition to being a trustee, she was an active supporter of Dana-Farber through annual fundraising events. She served on the executive, the science, and the gift planning committee, and was also a member of the Susan F. Smith Center for Women's Cancer Executive Steering Committee. And we have been and continue to be very grateful to Sandy, the Krakoff, and Coleman families for all that they have done for Dana-Farber over the years, and also very honored to hold this annual seminar in honor of Anna Maria Coleman. So as I said, this lectureship has been going on now for more than a dozen years. It really has allowed us to invite some of the key people in the field related to pancreatic cancer. If you go back more than a dozen years ago now and look at some of the lists, you see folks like Chris Iacobuzio and David Tuvison and Ben Stanger, Daphne Barsagi, Stephen Leach, among others. So really people that we all know well. And in that mold, it was really a wonderful opportunity to invite Alec to come back to Dana-Farber. I think as folks here may know, Alec did his MD and his PhD at Mount Sinai. He then did his residency in the Harvard Combined Radiation Oncology Program, and then did his postdoctoral fellowship in Ron DePino's lab. Fittingly, he was just here a few weeks ago and also gave a talk, which was also an exciting opportunity to have another Dana-Farber faculty member back. And then Alec was on faculty here at Dana-Farber. And about 8 years or so ago, he left Dana-Farber, went to NYU. At NYU, he currently is the Anita Steckler and Joseph Steckler Professor and Chair of Radiation Oncology. And he also, as of last year, is now also the Director of the Laura and Isaac Perlmutter Cancer Center at NYU Longhorn Health. So before I turn it over to Alec, really Alec has been a major mover in the field of pancreatic cancer, both him and his lab. He really has studied the metabolic needs of KRAS-driven cancers, pancreatic cancer in particular. He's made really seminal discoveries, he and his group, in a number of areas, including one of the first people to demonstrate the importance of autophagy in pancreatic cancer, how this relates to treatment resistance, immune evasion, and now really been working very hard to translate this to the clinic and bringing new therapies to patients with pancreatic cancer. He also has really been one of the major people in the field studying how KRAS is a regulator of metabolism, including glucose metabolism, glutamine metabolism, and how this metabolic plasticity is important as we define, again, new therapeutic vulnerabilities. And then the past number of years, also thinking how there is crosstalk in the tumor microenvironment at the metabolic level, including between stellate cells, neurons, tumor cells. So really, it's a tremendous honor for Andy and I to host Alec and for him to be here to talk with you all. We have a nice plaque here for him, and let's make sure he comes up and talks.
Thank you. It's really great to be back. I remember when this lecture started when I was here, and you had a lot of luminaries of pancreas cancer. Now you went down the list and you have me, but you know, you'll get back to the the good ones later. But it's really great to be here. It's great to see a lot of old friends, and it's amazing to see really what has happened here under Brian's leadership in terms of GI cancers and pancreas cancer. It's amazing. So I got a couple of disclosures. None of them are particularly relevant to this talk. So since most of you work on pancreas cancer, we'll just skip most of this. But really, if I have a pointer here, oh, here. Can you see this? Oh, there's a pointer. That's really the statistic that sums up how bad we're doing in this disease. And when Brian and I first started, this was more than doubled. It was like 5%. So there's been progress, but there's a long way to go. And the philosophy we've had in my lab is that perhaps some of this therapeutic resistance points toward altered metabolism in these tumors. So it's fitting because my good friend Lou Cantley is sitting in the front row. So actually more than 10 years ago now, he and I, our labs collaborated, and we identified a novel metabolic pathway in pancreas cancer. And for about 20 years or so, we've been studying tumor metabolism in this disease. And we've learned a couple of lessons that are fairly straightforward, and one is which these have an incredible metabolic plasticity. So this is a pathway that my group and Lou's group identified. I'm not going to go through the whole thing, but it basically uses the carbon skeleton of glutamine in the form of glutamate. It goes through a series of mitochondrial reactions, cytosolic reactions, and its main function is to actually produce NADPH to keep redox homeostasis in the cells. And we showed genetically, if you block some of these enzymes, you can have some responses in pancreas cell lines and models. We were a bit excited when Calithera came out with a clinical grade inhibitor to glutaminase called CB839 that we could perhaps translate this. And Joe Mancius, who is a postdoc for myself and Wade Harper, did some really great work understanding how this inhibitor worked, or in fact didn't work. And what Joe found was that in a matter of hours, when you look at the metabolome, and this is just showing some quantitative proteomics, that the entire metabolome was rewired when you inhibited this enzyme glutaminase. And what that basically meant was that after 5 days of having a pretty profound growth suppression, the tumor cells just started growing at the same rate as they were before. So they can rewire their metabolism at an incredibly rapid pace. The other thing that we've learned is that these tumors have multiple mechanisms of metabolic plasticity, including metabolic scavenging. So work from Daphne Bar-Sagi, Cosimo Camiso, and others have shown that they can actually take up extracellular protein and degrade that in the lysosome to produce building blocks and metabolites. My lab has been studying, as Brian mentioned, a process called autophagy, which I'll talk a little bit about today. And that degrades, the cell can degrade itself, its own constituents to produce building blocks. And they even have found the abilities to have developed these cooperative metabolic sharing networks in the tumor microenvironment, where I'm going to spend most of my time today. But when I started here in 2007, we began studying this pathway called autophagy, which, as many of you are familiar with, is essentially self-eating or autophagy. And I'm not going to go through the regulation, but it's predominantly regulated by starvation. And cargo, which can be the whole organelles like mitochondria, damage protein, protein aggregates. They get enveloped in a double membrane vesicle called an autophagosome. This fuses to the lysosome, where contents can be degraded. They can be shuttled back into the cytosol for bioenergetic or anabolic reactions. There are some compounds, medications, in fact, that can inhibit this process, namely the anti-malarial chloroquine and its derivative hydroxychloroquine, which is a rheumatological drug, and they actually act at the level of the lysosome by inhibiting lysosomal acidification. So if you do have interesting findings in the lab, we had speculated it'd be possible to take these into the clinic. And it's not hard to imagine how autophagy can be important in physiological and pathophysiological metabolism, because depending on what substrate ends up making it into the lysosome, you can really pretty much fuel any pathway in central carbon metabolism. And beyond that, Joe Mancius, who I mentioned before, discovered a novel type of selective autophagy that regulates iron metabolism through the degradation of ferritin. So autophagy can touch metabolism at many different levels in cells. So more than a decade ago, we identified that pancreatic cancers actually have high levels of autophagy even when they're grown in full nutrient complement, which is typically not what is seen in a normal cell. This is a non-transformed pancreatic ductal cell. They have low levels of autophagy unless they're starved. So we began investigating what autophagy was doing in these cells, and we showed in a variety of model systems that either pharmacological inhibition with chloroquine or hydroxychloroquine had responses in xenograft or PDX models. This is showing you can recapitulate tumor responses in xenografts by knocking down key autophagy proteins like ATG5, and then we crossed the KPC model, the autochthonous pancreatic cancer model, to an ATG5 conditional knockout and showed that that prolonged survival. So this work basically showed us that autophagy has cell autonomous and tumor cell non-autonomous roles in pancreas cancer. As far as tumor cell autonomous roles, it's incredibly important for maintaining mitochondrial metabolism and mitochondrial homeostasis, in a large part due to its role in maintaining iron metabolism, as Joe discovered. And when you block autophagy in the tumor cells, mitochondria become less functional, you get decreased proliferation. But then in a non-cell autonomous fashion, there are multiple roles for autophagy inhibition that are anti-tumorigenic. For one is one of the metabolic cooperative networks in the tumor microenvironment. It occurs from the fibroblasts to the tumor cells, and that's dependent on autophagy in the lysosome. So if you block that, you block this nutrient crosstalk. And then what I'm going to show a little data about, you basically can reprogram the immune system to attack the tumor cell. So together, these cell autonomous and non-cell autonomous mechanisms provide a strong anti-tumorigenic effect of autophagy inhibition. And a number of years ago, we showed that one of the mechanisms of how pancreas cancers evade the immune system is actually through autophagy. And what we noticed was, in a collaboration with Rushika Pereira's lab, was that in normal cells or non-transformed cells, MHC1 is on the cell surface as you would expect it. In many pancreas cancer cells, it's in these punctate structures, which we eventually showed were lysosomes and autophagosomes. And we went on to show that if you inhibit autophagy in vivo, this is using a dominant negative system that we developed in mice, that you restore class 1 on the tumor's surface, it presents more antigen, you get a significant influx of cytotoxic CD8 positive T cells, and this is just shown here pictorially, and you get anti-tumor responses. And I mentioned that we have drugs like chloroquine, which are, albeit weak, they are autophagy inhibitors that can be used in patients. And we showed that if you take chloroquine and you combine it with dual immune checkpoint blockade, PD-1 and anti-CTLA-4, first we show that you get a modest inhibition of autophagy using an autophagy reporter, consistent with chloroquine's weak inhibition of autophagy, but you get a cooperative or synergistic anti-tumor effect by combining chloroquine treatment with dual checkpoint blockade. So this basically showed that in a normal pancreatic cell, part of the immune evasion is due to the selective autophagy pathway that we went on to biochemically characterize, and that the MHC1 is constantly degraded off the tumor cell surface. When you block the lysosome or block autophagy, you decrease this, you get more MHC1 on the cell surface, and more T cells come into the tumor and provide anti-tumor immunity. And what was exciting to us, and really what we think really suggests that we need better autophagy inhibitors, that there are now 2 randomized phase 2 trials published in the literature combining gemabraxane plus or minus hydroxychloroquine. This first one is in the resectable setting that shows that there was an increased response rate when patients were randomized to hydroxychloroquine. And what was exciting for us and consistent with what we showed in the mice is that the patients who had hydroxychloroquine had more T cell infiltration. And then the Stand Up to Cancer trial demonstrated a significantly improved response rate in the metastatic setting with the same gemabraxane chemotherapy. So we're hoping that more potent autophagy inhibitors will eventually make their way into the clinic. So what I want to spend most of today on is our work that Brian mentioned about targeting metabolic networks in the tumor microenvironment. And then with a little time at the end, I'm going to talk about approach to really more broadly target metabolism, which we think may have some traction. So as everyone in the room who works on pancreas cancer knows, one of the defining features of this tumor is its complex microenvironment. It's defined by this intense desmoplastic or stromal reaction. Pathologists will tell you most of the time when you look under the microscope, the minority of cells that you see are epithelial tumor cells, and the majority of what you see is extracellular matrix, inflammatory cells, fibroblasts, et cetera. And we went on to show that, as I mentioned before, that the fibroblasts can support some of the growth of the tumor by secreting the amino acid alanine, and then we went on to demonstrate that we can define the nutrient transporters that mediate this, and we're trying to target these now by doing pharmacological inhibition. But it creates this desmoplastic reaction creates this poorly perfused, extremely hypoxic tumor microenvironment with an altered nutrient content. And when Robert Bond came to my lab a few years ago, he became interested in another cell type in the tumor microenvironment, the neuron, and wanted to study the metabolic contribution of the nerves in pancreatic cancer. And this is an interesting area for many reasons. Seminal work from Tim Wong at Columbia and others have shown that the neurons, depending on the type, can influence tumors, either pro-tumorigenic or anti-tumorigenic. So you have sensory neurons and sympathetic neurons, which are pro-tumorigenic, cholinergic neurons, which are anti-tumorigenic. But Robert really wanted to, but most of that work had really focused on secreting growth factors out of the neurons that the tumor cells used to fuel growth factor signaling. But Robert speculated that perhaps neurons can fuel tumor cells under nutrient poor conditions that were seen in the tumor. And the reason why this is very attractive as a hypothesis was that the fibroblasts, which we showed entered these cooperative nutrient sharing networks, are actually in the same nutrient poor tumor microenvironment, where the cell body of the neuron is actually in the peripheral circulation in the ganglion, where it's bathed in nutrients, and it's the axon that grows into the tumor. So Robert hypothesized that perhaps the neurons acted like funnels, funneling nutrient from the peripheral circulation into the tumor through the axons. So the way he studied this was utilizing a microfluidic device where you can grow rat DRGs in these chambers. Then through a tract into the axons grow through these chambers, and hydrostatic pressure separates the fluid from these two different chambers, so you can see what's being secreted by the axons versus the cell body. And when Robert did this, and we focused on amino acids, he showed that there were many amino acids that were secreted by the neurons, or the axons. We were particularly interested in serine and glycine, given they're important amino acids both in normal physiology, but over the past 10 years have gained a lot of interest in tumors because they produce they fuel several metabolic pathways. They produce phospholipids, protein, of course, one carbon metabolism, which is important for redox homeostasis, nucleosides, et cetera. And most of the serine, if it's not taken up extracellularly, is produced by glucose through a series of reactions that I'll go into later. And what was interesting about this is, at the time we were looking at this, Karen Vousden and Oliver Maddox really had a beautiful paper showing that pancreas cancers using the autochthonous mouse model can produce their own serine, so we're not sensitive to low serine levels. But when Robert looked at human pancreatic cancer cell lines, it turned out that about 40% of human pancreatic cancers can't make their own serine and require endogenous serine. And this is just showing examples of two lines that don't require any serine and two lines at the bottom that require exogenous serine. And this is just showing when you go across a panel that about 40% of them can't produce their own serine. Now, what was interesting about this and how they can't produce serine is an easy question to answer, but the why is somewhat more difficult, and we don't have an answer. But the how is straightforward in that they don't have PHGDH. So all of the pancreatic cancers that cannot produce, that are dependent on exogenous serine, lack one of the key rate-limiting metabolic enzymes of serine biosynthesis, PHGDH. Many also have low levels of PSAT1, but all of them have absent PHGDH. And what was very important to us was when we looked at a large collection of human tumors by immunostaining, the exact same percentage of human tumors don't express PHGDH as what we saw in cultured cell lines. So this wasn't a phenomenon of just culturing tumor cells for decades in DMEM. So we went on to show that the neurons can actually support the growth of these tumor cells in low serine conditions using these same microfluidic chambers. It only mattered in the ones that could not produce their own serine. And then Robert went on to try and figure out why the PDAC cells require serine. And if you remember what I showed you initially is that these tumors have amazing ways to reprogram their metabolism. But not surprisingly, the one area that they can't get away from is that you require serine to make protein. So that wasn't that interesting, but as we dug into it, what became very clear was that there was a selectivity in terms of how this was regulated, meaning that there was selective translation of specific transcripts. So Robert went on to design this elegant reporter system and also confirmed it with RiboSeq, which is a way of sequencing where the ribosomes stall during serine glycine deprivation. And what he found was, to our surprise, that there was this incredible specificity where on TCC and TCT codons during serine withdrawal or low serine levels, the ribosomes stalled on these two codons specifically, while the other codons, they were able to continue to translate. And you can bioinformatically go through the genome and look to see which genes would be predicted to be affected based on the amount or the percentage of stalled codons versus not. And when you take the top 5% of each of these and you look at the genes that are impacted, the gene set that was predicted to be the least impacted, meaning they have the lowest amount of TCC or TCT, were secreted factors, and nerve growth factor was at the top of that list. NGF actually has something like 80 or 90% favorable serine codons, so it's not impacted basically at all when you starve the cells of serine. You can see, in fact, when you decrease serine levels with these three different serine-dependent cell lines, you can see you get a significant increase in the amount of nerve growth factor that was secreted. So we wanted to move to an in vivo system to see if this was happening in vivo. So we orthotopically implanted a serine-dependent cell line in the pancreas of immune-compromised mice, put the mice on either a standard diet or a serine glycine-free diet. Not surprisingly, tumor cells that require serine when you take serine out of the diet don't grow as well, and that's shown here at about a 50% reduction in growth. This appears to be proliferation defect or decrease in cycling shown by phosphohistone 3. We don't see a lot of cell death, that's cleaved caspase 3. But what was very striking is when you looked at the tumors of the mice that were on a low serine glycine diet, you saw a massive increase in innervation. This is shown by a 3D method of staining for neurons using PGP 9.5. You can see this tumor nodule here as all as this innervation, and it's quantified over many different tumors shown here. So it appears that it is operational in vivo, that when you put the mice on a low serine diet, you get more neuronal innervation. So we wanted to prove functionally whether or not this impacted the tumor growth. And we took advantage of a compound that many of the oncologists in the room are familiar with, LOXO-101 or larotrectinib. It's an NTRK inhibitor that basically is the nerve growth factor receptor inhibitor. And in cancer, in about less than 1% or so of human cancers actually have a fusion with this receptor that becomes a driving oncogene. So this drug is used in those cancers. We actually wanted to use it for its initial purpose, which was to block neuronal innervation. So we did the same exact experiment as I showed before, except this time half the mice got LOXO-101 and half the mice got a vehicle control. And first we wanted to see if this worked, if it worked in terms of on target. So again, when the mice are in a full diet, you really don't see very much changes in neuronal innervation. When you put the mice on a serine glycine-free diet, the neurons innervation goes up, and this goes all the way back down to baseline when the mice were put on the LOXO-101 compound. Similarly, when you put the mice are on a regular diet, LOXO-101 has a very modest effect. Again, when you put the mice on a serine glycine-free diet, the tumor's growth slows down, and this is further impaired by giving the mice LOXO-101, and this is just showing the proliferation follows the same trend. So Robert's model was that in the 60% or so of human pancreatic cancers that don't require exogenous serine, whether or not the tumors or cells are growing in areas of the tumor that have high or low serine, they're still able to proliferate. In the 40% or so that are serine dependent, in high serine conditions, they're fine. When they grow into areas of low serine, they begin to decrease their proliferation. The nerve growth factor gets secreted through the selective translational program. Neuronal innervation occurs, serine and glycine are secreted into the tumor, and the tumor could continue to proliferate. So to summarize this part of the talk, which was published, and I'm going to move on to unpublished data that works on this a little bit further. Peripheral neurons can secrete amino acids such as serine. They can metabolically support the tumors in nutrient-deprived regions. And although, as I showed you, in many cases they can rewire their metabolism, mRNA translation or protein synthesis is a bottleneck. They can't get around that. And what I think is extremely interesting, and this is Robert's focusing his lab on this regulation now, is this selective translation, meaning that there are differences in translation efficiencies against different codons. And this is something that's common in bacteria and has really only been described in a handful of cases and never before this for serine in eukaryotic cells. And what's really interesting is that it's another way to allow selective biological responses. So things like nerve growth factor have something like 80 or 90% favorable codons, but cyclins and DNA replication genes have the opposite ratio where they're mostly unfavorable, so it allows the tumor cell proliferation to slow down while nerve growth factor calls in for neurons to secrete serine into the tumor. So Robert's lab is working on how this mechanism occurs. So this work is unpublished and something we're excited about. We wanted to try and understand about some of these metabolic networks in metastatic disease, and for the oncologists in the room, while Christine Akabuzio-Donahue showed like a third of pancreatic cancer patients die of complications locally, the majority of pancreatic cancer patients die of metastatic disease, and the predominant or overwhelming site of metastasis is the liver. So, we had shown that fibroblasts and neurons have important roles in the primary tumor in terms of supporting tumor cells growth. But the questions when tumor cells metastasize to the liver, how do they gain metabolic support? And it's not hard to make a hypothesis when you look at what a liver metastasis looks like. It's basically a cluster of tumor cells surrounded by a lot of hepatocytes. So the likely candidate cell that might be cooperating with the tumor cells are the hepatocytes. So Kasuke in my lab did an experiment where he injected tumors into the splenic circulation to create liver metastasis, or did a sham injection. Then he isolated hepatocytes and then just did simple RNA-seq. And one of the most upregulated genes in the liver was PHGDH. And we confirmed that on the protein level as well. So why would that be important? I just mentioned that 40% of pancreas cancers lack the ability to make serine. So these tumor cells need a source of serine when they metastasize to the liver. So again, we took human pancreatic cancer cell lines that were serine dependent and showed that when you co-culture them in the absence of serine and glycine, you can support the growth when they're co-cultured with hepatocytes. We wanted to move to a system that was more physiological in terms of the tumor microenvironment. So we basically took a KPC B6 cell line and CRISPR'd out PHGDH, since we knew that was the causative issue with the human cell lines, and we can recapitulate what we see in the human cell lines. Again, when you don't have serine and glycine, the tumor cells don't grow very well. When you co-culture them with hepatocytes, you can restore that. So we wanted to see whether or not the hepatocytes can produce serine that the tumor cells took up. So we did co-culture experiments in Boyden chambers, and we labeled the hepatocytes with uniformly labeled 13C glucose. I mentioned that glucose is the predominant source of serine biosynthesis. And you could see in normal tumor cells that possess PHGDH, they can make serine. Is showing this M+3, which comes from glucose. When you knock out PHGDH, this goes away. But when you co-culture the cells with hepatocytes, you can get back this M+3 fraction, meaning that the hepatocytes biosynthesized serine and the tumor cells took it up. So we wanted to move in vivo and look at this. So we took the B6 cell lines that have CRISPR'd PHDDH, injected them into the spleen of mice that could make liver metastasis, and then looked at histologically. What was very interesting was that you saw this very interesting pattern. When you knock out, when you have wild-type tumor cells, the tumors express high levels of PHDDH, and the hepatocytes have almost absent PHDDH. And then in the most extreme opposite setting, when the mice are placed on a serine glycine-free diet and you use the PHDDH knockout cells, the tumor cells do not express any PHDDH, but the hepatocytes significantly upregulate that. And you can see it just quantified here, that if the hepatocytes have low levels of PHDDH, the tumor cells have high, and vice versa. And functionally, you can actually see proliferation was impacted depending on the distance of the tumor cells from the hepatocyte. So if you make concentric circles of around the tumor cells that were labeled here in green with a cytokeratin stain, and then you did Ki-67 staining, the tumor cells that were closest to the hepatocytes had the highest level of Ki-67 staining, meaning they're more proliferative, and this dropped as you move further away from the hepatocytes. So, we wanted to validate this in human patients because this is important to make sure that this is a phenomenon that's seen in patients. We collaborated with Tony Hollingsworth and the rapid autopsy program he developed at the University of Nebraska, and we got matched liver metastasis and primary tumor from pancreatic cancer patients, and we're able to demonstrate that similar to what we showed in the primary human tumors, a significant fraction, about a third of the patients have tumor cells that do not express PHDDH. About a third or a little bit more have high levels of PHDDH, and then about a third have intermediate levels. And what we saw was when you looked at PHDDH staining in the tumor and in the hepatocyte, you saw again what we saw in the mouse model, that if the hepatocytes had very strong or high positive staining for PHDDH, the majority of the tumor cells were negatively stained. And conversely, if the hepatocytes were weak or had no staining for PHDDH, the majority of the tumor cells showed PHDDH expression. So mechanistically, we wanted to understand how the tumor cell might be signaling to the hepatocyte to increase PHDDH levels. So we did some co-culture experiments in the Boyden chambers where we could separate the two populations and did RNA-seq, and showed that when you compare the PHDDH knockout to the wild type co-culture with hepatocytes, or the knockout with or without hepatocytes, one of the secreted factors that was the most upregulated or downregulated, depending on which way you were looking at it, was CXCL5. And not surprisingly, the hepatocytes showed changes in serine biosynthesis enzymes. So we wanted to functionally see if CXCL5 had an important role in vivo. So we again took the knockout in the wild type cells, injected them into the spleen, and then looked at the amount of CXCL5 by ELISA. And you could see that when the wild type tumor cells in the liver have very low levels of CXCL5, even in high and low serine conditions, when you knock out PHDDH, the metastases in the liver have very high levels of CXCL5, particularly when they're grown in serine glycine low conditions. We can actually look at this in patients because we were able to have frozen either match normal liver or liver metastasis from these patients. And again, the patients with liver metastasis had much higher levels of CXCL5. And because we were able to do IHC on each of these patients to see we knew the PHDGDH status of each of these patients. And if you look in this population here, if you had high levels of PHDGDH GDH expression in cancer cells, the amount of CXCL5 you had was low, and if you didn't have PHGDH, you produced significantly higher CXCL5 in patients. So we can say that this was also occurring in human patients. Functionally, if you take we wanted to see how this was regulating PHGDH. So hepatocytes alone have very low PHGDH expression. When you co-culture them with the knockout cells, you can see a significant induction of the PHGDH transcript. And when you knock out CXCL5 in the tumor cells, you lose this upregulation. So this was a CXCL5 dependent phenomenon. So we CRISPR'd out CXCL5 in the PHGDH null cells, injected them into the spleen, created liver metastasis, and you can see there was a significant decrease in metastatic burden as measured by liver weight when you knock out PHGDH, but also looking at tumor histology by quantifying how much area was metastasis. So CXCL5 is important for the growth of these liver metastases. And you could see when you silence CXCL5, you decrease the PHGDH expression in hepatocytes. So here this is CXCL5 wild type cells. You can see you have high levels of PHGDH in the hepatocytes, which are the non-labeled cells. This is a cytokeratin staining. When you knock out CXCL5, this staining essentially goes away, and that's quantified over here. And similarly, proliferation follows the same pattern, that when CXCL5 is knocked out, you get a significant decrease in proliferation of the tumor cells. So what I've shown so far is that pancreatic cancers that can't produce their own serine, when they metastasize to the liver, secrete CXCL5. This binds to its cognate receptor CXCR2, which I'm going to talk about in a minute. This upregulates PHGDH expression. It makes serine. The tumor cells can take up the serine and grow. We have inhibitors to CXCR2, which is the receptor to CXCL5, that are being used in clinical trials. So we wanted to test what these were doing functionally. When you treat co-cultures with the CXCR2 inhibitor and then look at the expression in the hepatocytes, you see a downregulation of the entire serine synthesis pathway, consistent with what we showed before with CXCL5. So we moved in vivo, where we took the serine-dependent cells, injected them again into the spleen to create liver mets, and then randomized mice to get CXCR2 inhibitor vehicle control. And this is an extremely complicated graph because we did a lot of controls, so just trust me, I'll point to some bars here that are important. When you knock out PHGDH, as we showed before, you get a decrease in liver metastasis in these tumors in the serine glycine low conditions. When you add the CXCR2 inhibitor, this almost completely abrogates liver metastasis in these tumors. Once again, we showed that this has an impact on the ability to produce labeled serine. So again, this is tumor cells that can't produce serine without hepatocytes. They don't have any or very low M+3 labeled serine. When you co-culture them with hepatocytes, they're able to take up the M+3 labeled serine that the hepatocytes produce. But when you treat them with a CXCR2 inhibitor, you lose the labeled serine. So functionally, this is critical for serine production and then uptake by the tumor cells. So to functionally prove that the PHGDH upregulation in the liver was critical to support the growth of metastasis, we used a model that was made by Ben Stanger's lab, which essentially is a LSL rosa Cas9 mouse, and then you have a adenovirus that has a guide RNA. In our case, it was the PHGDH. It has a liver-specific promoter that drives a CRE. So basically, you could selectively knock out PHGDH in the hepatocytes. So we first tested this, and you can see when you looked at purified hepatocytes, you get a significant reduction of PHGDH expression in the adenoviral injected mice. So we then performed experiments to look at liver metastasis. So first, when you take a control guide and you inject PHGDH null tumor cells, you could see that you get a massive upregulation of PHGDH expression in the hepatocytes. When you knock out PHGDH, you get a reduction in the basal expression, not surprisingly, of PHGDH in the hepatocytes, but also when you put the mice on a serine glycine null diet and inject them with the PHGDH null tumor cells. So functionally, we're able to decrease the expression in the liver, as I showed you before. And importantly, this led to a significant reduction in tumor growth, shown by tumor weight, but also by tumor area. Knocking out PHGDH into tumor cells significantly reduces tumor burden, but when you knock it out in both the tumor cells and in the liver, you most significantly reduce the growth of liver metastasis. So the model is that in the 40% of pancreatic cancers that can't make their own serine, when they metastasize to the liver, they then secrete CXCL5, which binds to CXCR2. It increases PHGDH expression. They make their own serine. The hepatocytes make serine and feed the tumor cells so the tumor cells can proliferate, and you can block this at various levels by blocking things such as CXCR2 with either genetically or by pharmacological inhibition. So in summary, hepatocytes can secrete serine. They can support pancreas cancer metastasis, growth, and survival. The tumor cells signal to the hepatocytes through the CXCL5, CXCR2 axis to upregulate PHGDH. Critically, human tumors show a very similar pattern of reciprocal PHGDH expression in tumors and hepatocytes. And we think that targeting hepatic serine biosynthesis by interfering with PHGDH expression when you inhibit CXCR2 can decrease liver metastasis burden. There are now companies that are entering early phase clinical trials with serine glycine free or low serine glycine diets. So there may be an interesting opportunity to combine dietary therapies with CXCR2 inhibition in pancreas cancer patients. So with the few minutes I have left, I want to sort of talk a little bit about something that we're pretty interested in, which is, targeting metabolic pathways using a more broad or blunt instrument. I mentioned, for a lot of years, including the work we did with Lou and many other things, we were trying to find very subtle changes in tumor metabolism that were specific to the tumor, target those, and see if that produced anti-tumor effects. But as I showed you, the tumor cells always found a way to rewire their tumors, and there were exceptions like things like serine, because even if you can rewire your whole metabolism, you need it to translate protein. But for the most part, the tumor cells are able to find their way around it. So a talented grad student in my lab, Yoel Encarnacion Rosado, wanted to look at what happens if you just did this more broadly and you just bluntly inhibited large swaths of metabolism. What would happen? So I told you about that work earlier we did with glutamine metabolism. And in that pathway, we inhibited the carbon skeleton of glutamine, which is just one aspect of its role and not any of the others. So Yoel wondered what would happen if we inhibited all enzymes in the tumor that utilize glutamine. And believe it or not, this was actually tried in patients a few decades ago with a compound called DON, which is essentially a glutamine analog that irreversibly inhibits every enzyme that utilizes glutamine, depending on the concentration and the KMs. Not surprisingly, it was toxic, particularly caused GI tract and liver toxicity, so it was largely abandoned. However, Drayson came up with a modification of DON, and they essentially esterified it and did a couple of other things that they proposed more selectively allowed release of the DON in the acidic tumor microenvironment that had higher levels of esterases. And we were somewhat skeptical of this, but we got the compound and proceeded to use it. And not surprisingly, DRP-104 and DON, which are the same, create, the only thing we could really characterize this as a metabolic crisis. You couldn't really characterize individual metabolic pathways. But importantly, DON and DRP-104 looked very similar in terms of the metabolic catastrophe that they were causing in these tumor cells. So Yoel moved to a syngeneic orthotopic model, and he implanted tumor cells into the pancreas and then randomized mice to get DRP-104 versus vehicle. And the mice actually did fine. There was very little weight loss. But surprisingly, this was the first time that we had ever used a metabolic inhibitor that we saw single agent activity, and this is in 2 different B6 cell lines that we implanted into the pancreas. It was stasis. It wasn't tumor regression, but it was fairly significant. And this is both in the primary tumor and in the liver metastasis model. Now, one of the main indications that this drug was going to be used at was actually in the IO space. There was some data that using this glutamine antagonist more selectively perturbed like MDSCs and other suppressors of the immune response, so that was thought to be able to enhance checkpoint blockade in a variety of tumor models, like the classic B16s, MC38s, et cetera. Interestingly, and not surprisingly, pancreas cancer behaves differently. The anti-tumor activity does not require the immune system. So you can see the exact same effects that we saw in the syngeneic models in nude mice and NSGs. So this does appear to be a tumor intrinsic effect, a true metabolic effect. And we also tried it in a number of PDX models that we had available to us at NYU, and we saw anti-tumor activity pretty much across the board. So broadly inhibiting glutamine metabolism creates a metabolic crisis. You can decrease the proliferation in vivo and in vitro. I didn't show the data, but one of the compensatory mechanisms is that MAP kinase is upregulated, and you can actually co-target MAP kinase and DRP and glutamine metabolism with DRP-104 and further prolong survival. But I think this is an interesting concept because it sort of says if you can have some more specificity in terms of drug delivery, you can potentially more broadly target tumor metabolism without toxicity. And this drug is in phase 1 clinical trials, and I believe it's being fairly well tolerated. So there may be hope of more broadly targeting tumor metabolism. So I'd like to thank all the people in my lab who did the work. The autophagy work was done by many, many people over many years. I mentioned the collaboration with Rashika's lab for the MHC1 stuff. The glutamine, the DON DRP was done by Yoel Encarnacion, who now is doing a postdoc at the Salk. Colina Ganguly did the liver metastasis work that I showed, and I mentioned Robert Bond's work for the neurons. These are my funding sources, and I can stop here and take any questions.
Very nice presentation. Very much enjoyed the talk. I was just wondering if serine metabolism is required just for liver metastasis or also for the lung metastasis as well.
So the question is whether serine metabolism is required for lung metastasis also?
Yep.
I mean, presumably, we don't know what this group of tumors, how they get serine from in the lung. It's obviously not hepatocytes, but we haven't tested it. But presumably, they're going to need a source of serine because they can't make it themselves. So they probably co-opt.
Some tumor innervation or something.
It could be
like,
innervation.
Yep.
It could be pneumocytes.
Yep.
We're going to do that. We haven't looked yet.
Thank you so much.
Yeah.
Thank you so much for your talk. Do you have any clues if this serine is preferentially used for some proteins once it's supplied to the pancreatic cancer cells, so some proteins are preferentially synthesized rather than others, or not?
So the question is whether there's a preferential utilization.
Exactly.
Yeah, I mean, because there's a preferential difference in the translation rates, our presumption is that it allows the ones that have essentially stalled translation to continue being translated. But to be honest, we haven't done a real thorough job in a time course looking at that. But the ones that are predominantly with favorable codons can still be translated. So we presume it's for the non-favorable codon-containing proteins, which mostly fall in the realm of proliferation, cell cycle, and allow restoration of proliferation.
Hey, very nice talk. I have questions about serine metabolism and glutamine metabolism part. So for serine metabolism, my question is that what about the does autophagy plays any role in supplying serine, particularly because when cells are under migration or invasion during the metastasis process, they don't have any other probably cells to supply serine. So in that case, could autophagy be helpful?
It's a good question. I mean, I would say globally, one of the interesting things was that there appears to be specificity in terms of what metabolites autophagy is providing. We don't really see an upregulation of autophagy in the serine deprivation setting, but you mention a very specific context, which you're talking about when they're sort of traveling through the bloodstream, and we haven't checked that. So it's possible that you could get a temporary upregulation of autophagy to allow them to see in the liver. But we haven't looked at that. It's a good point.
Thanks for the great talk. Just curious about whether you had any hypotheses for determinants of CXCL5 expression and also kind of determinants more broadly of exogenous serine dependent versus independent PDAC cells. Is it cell state or is it genetic determinants? And also, it presumes maybe there's a sensor for serine that upregulates CXCL5.
So to answer your question about, the PDAC cells and why they lose serine, or if there's a correlation with serine biosynthesis, we don't really know. I mean, it's not genetic, meaning the locus is intact. It's probably epigenetic, but what's odd is it's completely irreversible. So you can take these tumor cells and Robert grew them in, decreasing concentration of serine for months, and he was never able to get them to upregulate PHGDH. As to why, we've speculated a number of things that, it's all hand waving, but, if you have serine, perhaps you don't need to make it, obviously, although one would think that, the feedback mechanisms of metabolism would be more applicable or relevant to that, right? So if you have serine, you could downregulate temporarily the expression of PHGDH, but this is permanent. So we don't know why. As far as the CXCL5 upregulation, that in terms of what's driving it, we don't know yet.
Hi, great talk, thank you. So I was curious about other cell types that may also be affected in this. So you're looking specifically at the CXCL5 and CXCR2 linkage between hepatocytes and tumor cells, but obviously in the tumor you're looking at nerves as well, but there's fibroblasts, there's other immune cells. Have you looked to see what other cell types would make CXCR2, or would, could this same mechanism of serine upregulation occur in fibroblasts and actually support that growth in, the liver or in the immune cells that are infiltrating that metastasis?
So there's a couple of parts of that question. We did check fairly carefully as to whether cancer-associated fibroblasts can produce enough serine to support tumor cell growth, and they don't. As to whether that's due to CXCL5 secretion, we're working on that now to try and understand whether that's the mechanism or whether some cells express the receptor and some don't. I think that's an important question that we should have an answer to soon. I suspect it's going to be some combination of their inability to upregulate CXCL5 or not have the receptor.
And for the immune system? Have you looked at the immune cells?
In terms of what they're doing with this pathway? No. I mean, it's very possible, as you know, that the CXCR2, CXCL5 axis is important in the immune system. I can tell you that if you what we're going to do now is we're going to do the same experiment using Ben's liver mouse, and we're going to knock out CXCR2 in the hepatocytes to go one step upstream to see if we can get the same effect that we did when we knocked out PHDDH. So that won't tell us that the immune cells are not getting that signal, but it'll tell us whether or not that's important for tumor growth. So we're doing that experiment now. Thanks.
Sorry, my question is really related to the previous question, and you almost answered, but I'm curious about the CXCL2 expression on neutrophils or like granulocytic MDSCs, because as you know, that this is well-known chemokine receptor for neutrophil migration. So just wondering whether, like in like setting depleted condition, either like influx of neutrophils or MDSCs, which could possibly suppress the anti-tumor immune responses.
It's a great question, and we have the tumor sections. We should probably do that. We have not looked at the immune infiltrate in that setting, but it's a great experiment and easy to do.
Thank you for your great talk. Yeah, I have a brief question. In your previous data, there is KRAS is associated with metabolism as well. So, do you have any evidence between the synergism of other chemo agents or targeted agents like gem-Abraxane, FOLFIRINOX, or KRAS inhibitors?
KRAS inhibitors in which setting?
In association with serine metabolism. Yeah.
No, we haven't. I have a med-onc fellow in my lab, and we're thinking about things to combine serine deprivation with. In the trial that Fife is sponsoring, they're combining it with chemotherapy. So we'll get an answer in patients as to whether or not, well, one, they can remain compliant with a serine glycine-free diet, and two, whether there is some interaction with chemotherapy.
I was just also curious if loss of serine or knockout of PHGDH caused some phenotype in the liver or some sort of like changes like, or metabolic rewiring. Like, is it like when there is loss of serine in the liver or the tumor, like there's some sort of serine adaptation or maybe some sort of other like metabolites, maybe fatty acids? Like, do you see that? Like, given that this is so plastic, like metabolic rewiring?
See, the only thing, it's a good question. We haven't done detailed metabolomic studies. We've done sort of very basic liver function characterizations, and the LFTs look pretty normal. So it doesn't look like they're hurt or impaired in any way. But it very well could be due to the fact that there's a metabolic rewiring in the hepatocytes.
So as glutamine is quite important for nucleotide synthesis, I'm wondering if a subset of cancers which have mutation in DNA repair pathways, are they more sensitive to glutamine inhibition?
We haven't tested that, but it would be a very reasonable hypothesis.
Excellent. Thank you. You want any more? All right, just under the wire, make it fast.
Sphingomyelin metabolism is serine is a precursor of sphingomyelin, and it's turned over primarily in lysosomes. Is there a relationship there between the serine minus phenotype and the autophagy phenotype?
There could be. I mean, the only thing I can tell you is that when you take away serine and glycine from pancreatic tumor cells, they can maintain all of those metabolic pathways in a way that doesn't impair their growth. It doesn't mean that they're not being stressed in a way where that's relevant, but we didn't see changes in sphingomyelin metabolism.
Excellent. Please give out to the